Professor Fahal, do you think we should start now?
Yeah, yes.
Okay. Hello, everyone, and welcome back again to the Mycetoma Research Club. And in today's session, we will be having Dr. Rami Ahmed. He will take us through nanomedicine for treatment and prevention of mycetoma disease. And I would like to handle it to you, Prof. Fahal.
Thank you very much, Dr. Rami, for giving us the opportunity to talk to you and to listen to you. Dr. Rami is an eminent teacher, pharmacist, and a scientist. Currently, he's in South Africa doing his postdoctoral training and activities there. In nanoscience, it is a very exciting area for research in mycetoma and for other neglected tropical diseases. We are looking forward for a nice, elegant presentation on the use of nanoscience for the treatment and prevention of mycetoma, a disease which is badly neglected both locally and internationally, affecting the most unprivileged communities around the globe. Mr. Sami, the floor is for you. I'm looking forward to listening to you, and we have a fruitful discussion at the end of this presentation. Thank you very much.
Okay, thank you, Prof. Fahal. Can I start now?
Yes, please do.
Okay, good afternoon, everyone. I would like to sincerely thank the Mycetoma Research Institute Journal Club for giving me the opportunity to speak with you today. It's a privilege to share insight on nanomedicine, its growing importance in the treatment of infectious disease in general, and its promising potential as a strategy to combat mycetoma in particular. My name is Rami Ahmed. I hold a PhD in pharmaceutical science from the University of Western Cape. And as Prof said, I'm a postdoctoral researcher now in infectious disease nanomedicine lab, where my research focuses on developing advanced drug delivery system to improve the treatment of challenging infections. So I'm excited to take you through the today's presentation and explore how nanotechnology can help address some of the key challenges we face in mycetoma therapy. So my outlines is introduction to mycetoma and nanomedicine, challenges in the current mycetoma treatment, the role of nanomedicine in drug delivery as general, and the current application of nanomedicine in infectious disease, and some research gaps and opportunity in mycetoma nanotherapy. So all of you guys know that mycetoma is a chronic, progressively destructive infectious disease caused either by the fungi, the Mycetoma, or the bacterial, the actinomycetoma. The mycetoma present a significant treatment challenges due to the deep tissue penetration of the pathogens, the fibrotic lesions that formating the matrix structure around the pathogens. So these complex grain structure prevent the permeation of most antimicrobial, if it is antibiotic or the antifungal. So in nanoscience, nanoscience as a general terminology is a study of the structure and material at the nanomaterial scale. When we applied these principle in various industries like the electronics, the textile, the mining, and the health, it's called nanotechnology. And when we apply this principle particularly in health and medical purposes, it's called nanomedicine. So the nanomedicine, the application of nanotechnology for medical purposes, we see that there is a potential to revolutionize mycetoma treatment, diagnosis, and prevention. There are various challenges in the current mycetoma treatment. These challenges can be summarized in 8, or some of them they are here. The limited effective treatment, some reports saying that some of the antifungal, they have a limited efficacy, and most of them, they are fungistatic, not a fungicidal, and they need a long duration for treatment of 6 to 12 months. This long duration can raise the problem of toxicity and the compliance of the patient. Also, sometimes there is a need for surgical intervention, uh, which is unfortunately, some of these facilities are inaccessible in many endemic areas. Also, there are some lack of early diagnosis because the disease is painless progression. Also, sometimes the misdiagnosis of the mycetoma because the mycetoma can resemble other chronic skin infections or tumors. Uh, Also, mycetoma is one of the neglected uh, disease, neglected tropical disease by the WHO that received limited funding, research, and public health attention. Beside that, also till now, there is no current or prophylactic measure for the disease itself. And lastly, and the most important, is the drug delivery barriers that is mainly coming from the grain structure of the pathogens that is formulated around the pathogens. So my presentation will mainly focus on the drug delivery and the problem that is related with the delivery of the antimicrobial to these pathogens. This will guide us to look behind the grain structure. As we said, the mycetoma, either it can be bacteria or fungal infection. There is a slight difference between the bacterial and the fungal grain structure. Understanding this structure of the grain can also help us to develop this nanomedicine. Generally, the grain, as we said, it can be a barrier that prevents most of the antimicrobial penetration to the pathogens, because these grain composed of multi-layers. Mostly it's 4 layers, starting with the core that accommodating the pathogens. If it is the bacterial or the fungal, there is a slight difference in the core between these two microbes. In the bacterial, for example, it can be like a filaments, compact, while for the fungal, it's a cement-like structure, because the fungus can release some of proteins and polysaccharides that can form a hard core structure of the grain. Then we have the peripheral layer. This peripheral layer is mainly composed of the neutrophils. Then we have the host reaction layer that is rich with the macrophages, and lastly, the inflammatory zone, which is enriched with the lymphocytes. So we think one of the hurdles that can face the delivery of most of antimicrobial is to how to penetrate these grains, especially the fungal one, because it's hardly structured, and as we said, in the matrix formulation. So nanoparticles, as a definition, nanoparticle is a microscopic particle with at least one dimension between 1 to 100 nanometers. 1 to 100 nanometers, for example, if you see this scale, 1 to 100 nanometer is, for example, it's 100 times less than the RBC's size, or 1,000 less than the width of a hair. So once we say that at least one of their dimension has 1 to 100 nanometers, because sometimes you can find other shape of the nanoparticle with multi-dimension. But for the spherical one, for example, we can define this nanoparticle with one dimension, which is 1 to 100 nanometers. This type of material, they exhibit a unique physicochemical and biological properties, which is mainly due to the larger or due to the large surface area to volume structure. This large surface area to volume structure means that we have a greater proportion of the nanoparticle atoms and molecules exposed on its surface. So meaning that there is high possibility of surface interaction, high possibility for drug loading and release, high possibility for reactivity and bioavailability beside various cellular uptake due to the interactions. The classification of the nanoparticle is mostly based on the composition of the material itself. They can be classified as lipid-based nanoparticles such as the liposomes or solid lipid nanoparticles, or it can be composed of polymers that is called the polymeric nanoparticles. Some of them, they are coming from natural source like the gelatin and alginate, and some of them, they are synthetic like the polycaprolactone nanoparticles or polycaprolactone polymers. Beside that, also we have the metal type of nanoparticles such as the silver, the gold, the quantum dots, and so on. The sensitivity of nanoparticle itself, there are two main approaches. Either we can start from the top to the down approach, meaning that we can start with macroparticles in millimeters or micrometers range, and then we can introduce different energies to decrease this sizing, such as applying the ultrasonic energy to decrease or to do a milling for these nanoparticles, or we can use the mechanical force to decrease this size. Or at the same time, we can start from the bottom, which is the bottom-up approach, which meaning that building the nanoparticle atom by atom or molecule by molecule from the smaller unit to achieve particular size range. By using, for example, different techniques such as the chemical precipitation or cold precipitation or the green synthesis. During the synthesis itself, there are various parameters that you can work on it to achieve different shape, different size, different characteristics such as the porosity, different responses to the external stimuli. You can produce nanoparticles that can have some responses to the pH, term of conformational changes, or temperature, or the light, or the thermal. At the same time, you can engineer the nanoparticle itself at the surface to achieve, for example, some targeting behavior by conjugating some antibodies or targeting proteins. Or sometimes you can conjugate some of the polymers at the surface of this nanoparticle to enhance the properties or the bioavailability of the nanoparticle itself. As application in their biomedical application, there are various applications such as the drug delivery, imaging and diagnostic application beside the vaccine and immunotherapy, and organ tissue engineering and wound healing. In the drug delivery, we have some problems or some limitation by using the classical or the conventional system. These limitation can be summarized as bioavailability or pharmacokinetic problems, such as the solubility problems or the stability of the materials or the therapeutic agents. Also, we have related with the classical dosage form, which is coming from the non-selective distribution of the therapeutic to the healthy tissues, beside the unfavorable dosing regimen by, for example, using high dose of the drug uh, for long duration, such as for the chronic diseases such as in tuberculosis or the mycetoma itself. We think that nanoparticle drug delivery system can be engineered to solve these issues. Typical architecture or structure of the nanomedicine or the nanoparticle drug delivery system composed of the carrier itself, which is the nanocarrier, if it is lipidic nanocarrier, the polymeric or the metal one. The drug or the therapeutic agent can be encapsulated within the nanoparticle itself, or it can be coated or attached to the surface of the nanoparticle based on the characteristic of the drug and the characteristic of the nanocarrier. Also, you can attach some of the targeting ligands, such as the polysaccharides or the antibody, to achieve the targeting behavior. And at the same time, as we said, you can sometimes coat your nanoparticle with polyethylene glycol or some polymer to achieve some desired characteristic to enhance the bioavailability of the nano formulation. So the general role for the nanoparticle in drug delivery system is to achieve the drug in the right place at the right time with the right dose. This achievement can give a safer medication because you are targeting your drug to particular region.
لا تكون مرة واحدة في حياتي تعملها. والله أنا الأسابيع اللي فاتت دي كلها بعملها دي ما ممكن يا أخي.
Please mute yourself, please. Please mute yourself.
To achieve the safer medication. To achieve the safer medication because you are targeting particular tissue or organ within the body and not the healthy one. At the same time, achieving a targeting behavior, meaning that you are accumulating your drug at the site of action, so more interaction with the pathogen or with the disease itself. When we see the application or the discovery of the nanomaterials from the first report of the technology itself as a nanoparticle, then the application of the clinical trials for them as a nanocarriers. And some of them, as you can see from this slide, some of them, they are already FDA approved, such as the Doxil, one of the anti-cancer that is based of using the liposomes as a carrier for doxorubicin anti-cancer chemotherapeutics. Also, one of the recent application for using the nanoparticles during Corona, the COVID-19, all of you guys heard about the Pfizer vaccine or the Moderna vaccine, and both of them, they are based on lipid nanocarrier for the messenger RNA, because as you know, the biological material, they are sensitive and they can be hydrolyzed inside the body beside their poor permeability due to high molecular weight. When we screen the nanomedicine in the clinical trials, we see that there is a primarily focus of the application of nanomedicine on cancers beside the other diseases. For example, the cancer represents 56% of the clinical trials that is currently based on the nanoparticle or nanomedicine, beside the other diseases, and the infections or the infectious disease represent 15% of that. These trials, they are approximately 600 to 700 trials till December 2024. When we focus on the infectious disease and after the COVID-19 pandemic, we see most of these trials, they are focusing on the COVID and other viral disease. And when we see the fungal only represents 7% of the trials, which is approximately 8 to 10 clinical trials that are using the nanoparticle system. And most of these trials, they are in phase 1 or phase 2, and they are using the ribosome or the protein nanocarrier. For the infectious disease, some of these drugs are already approved for the use. Till now, there are approximately 9 approved FDA nanoformulations, and 3 of them they are using or they are indicated for treatment of fungal infections, and they are using the amphotericin B via intravenous way, and none of them they are focusing for mycetoma treatment. We think that the application of nanomedicine in mycetoma can solve some of the problems related with the treatment. For example, the poor drug penetration inside the grain or the toxicity that is related to non-selective administration and the long duration of the treatment can be alleviated using the nanomedicine, since some theories have verified that the nanoformulation has the capability to penetrate even within some of the fibrotic tissues. Behavior of this nanoformulation can reduce the side effects, so safer medication, and also you can control the release of these drug from the carrier, so you can achieve a sustained release behavior and better bioavailability. So there are some gaps application of nanomedicine for the mycetoma treatment. I have summarized some of them. The first one is the lack of targeted drug delivery system, and currently, especially for the fungal one, they are rely on long-term antifungal therapy, and often they are toxic and poorly effective. So there is a need for targeted delivery to infected tissue. Also, there is no clinical validated nanocarrier for mycetoma. We think that the use of nanomedicine or nanoformulation beside the potential ligand for this nanoparticle can be used to target the mycetoma grains via different approaches. We can target the fungal grain component, especially, for example, for the fungal type. We found some of the proteins, they are rich within the matrix. For example, the fructose biphosphate aldolase, the GAP-DH, the enolase. These proteins, they are bind with the extracellular component like the laminin, the collagen, the plasminogen, the fibrinogen, and the fibronectin. We think that these nanoparticles functionalize with the ligand or antibodies to target these proteins or their binding partners, like the laminin and collagen, could improve the selective delivery to the fungal grains. Also, we can target the melanin biosynthesis pathway or the melanin itself by using the melanin binding ligand or melanin inhibitors, such as the tricycladol, which can help to disrupt the protective layer and enhance the drug delivery. Also, another way is to target host drive component in the grain matrix. As we said, the matrix can contain the collagen and the laminin. So the nanoparticle conjugated with ligand that bind to this extracellular matrix protein may facilitate the accumulation at the grain surface. Also, the matrix metalloproteinase, the MMP, especially the MMP9, is involved in capsule formulation. So the incorporation of doxycycline with a nanoparticle or the ligand to target the MMP-S9 could use to disrupt this capsule and enhance the drug penetration. Also, targeting the vessel's transport and the secretory pathway proteins, which is involved in the fungal vesicle transport, such as the sortilin and clathrin. They are implicated in the gray matrix formulation. So we think nanoparticles designed to interfere with this pathway or deliver inhibitors could prevent the grain formation or destabilize the existing grain. And lastly, the immune system modulation. So, like for example, designing nanoparticle with antibody fragments or peptide that to bind the bacterial grain, so to enhance the bacterial targeting and improve the host direction or the host efficiency. We think, as we said, the nanoparticle can be engineered to bypass the biological barrier that is formulated by the pathogens and deliver the antifungal directly to the grains. So as you know, there is not yet tested in mycetoma, but there is similar strategies have been succeeded in tumor models with the fibrotic stroma in cancers, and at the same time with the TB granuloma, because there is some similarities between the TB granuloma and the mycetoma grain. Yes, both of them, they have the pathogen within the core, and then you have multi-layers of the immune interaction or host interaction layer between the pathogen and the immune system. And some of these reports in the literature have verified that targeting the TB pathogen inside the granuloma can be achieved using different formulated or different engineered nanoparticle, and they have confirmed that the MTB bacilli within the macrophage have been destroyed using these systems. So we think designing of these nanostructures composed of any antifungal or antibacterial drug with some ligands that can inhibit or interfere with the formulation of this grain can improve the drug delivery and enhance eradicating of these pathogens. And as you know, most of these antifungals, such as itraconazole or the currently enter the clinical trials, the new forzafoconazole, they hinder or they have a challenges of poor grain penetration. So this system can interfere with this grain and can deliver these therapeutics directly to the pathogens. Also, other lack area is the limited preclinical and clinical studies. There is scarcity in vivo studies testing of nanomedicine formulation. Also, there is no clinical trials conducted for mycetoma. So there is a need to evaluate the safety and efficacy for these systems. Also, there is a poor understanding for the pathogen nanoparticle interaction, if it is via the permeability or the direct interaction of this system with the bacteria or the fungal pathogen of mycetoma. The vaccine also, it's a gap area because nanomedicine is widely explored for the other infectious disease and neglected tropical diseases, but its application in mycetoma vaccine has not yet been realized or documented in the literature. Also the diagnostic part, because the diagnostic, if it is the misdiagnosis or the early diagnosis, is one of the challenges that is related with the disease itself. So we think the nano diagnostic could improve early detection. It's crucial for the effective treatment. Till now, there is no developed or validated nanobiosensor nano-imaging tool for the mycetoma. So as a conclusion, addressing these gaps is crucial for the effective mycetoma treatment and prevention. Future research should focus on targeting delivery, the preclinical and clinical studies, pathogen, nanoparticle interaction, mycetoma vaccine, and diagnostic nanotechnology. Thank you for your listening.
Thank you very much, Rami. This is really a very fascinating presentation, a very fascinating ideas, and a very fascinating pathway for the future. I think is the sky is the limit to think about how can we improve the quality of the life of the affected patient with mycetoma and other neglected tropical diseases. You had opened our eyes in a new venues and avenues on how we can structure our research and to design clinical trials addressing the different aspects of mycetoma, including therapeutic, diagnostic, as well as a prevention measurement for this neglected ugly disease. Again, thank you very much, and the floor is open for discussion.
Thank you so much, Dr. Rami, for the eye-opening session. I have a question. Does the molecular weight of the drug affect designing of these nanoformulations? Because itraconazole has a molecular weight, I think, of 705, and the phosphoraboconazole has a molecular weight of 547. So does these molecular weights affect the designing of these drugs in nanoformulations?
Okay, thank you for your question. The size or the molecular weight of the drug itself, sometimes it can affect the properties of the nanoparticle, but it will not extremely affect the overall nanoformulation, because as we said, various biological materials, as you know, the proteins, they characterized by a very large molecular weight of 100 or 200,000 kilodaltons. They can be loaded within these nanoparticles, and they can be applied to achieve various organs or tissues within the body. So they can be achieved, even, for example, compared to the biological. I think the ketoconazole and the itraconazole, they don't have a very big molecular weight compared to the various biological materials.
Okay, thank you for this. If anyone has a question, you can raise a hand. Dr. Ali, yes, go ahead. Dr. Ali, you're muted.
Yeah, sorry. Hello, everybody. Thank you very much, Dr. Rami, for this nice and fruitful presentation. As Profahal said, it opened our eye to an area or a new area. I have a comment because nowadays maybe AI and machine learning are rapidly advancing and even transforming many areas of healthcare and even pharmaceutical developments. And this AI and machine learning technologies have the potential to optimize the design of nanoparticles by predicting their behaviors, improving drug loading efficiency, and I think even in the tailoring service properties for targeted delivery. My question is there any possibility, or how might AI or machine learning be integrated specifically to optimize a nanoparticle design for all neglected tropical disease like our mycetoma, and where research resources are often limited in this area. Thank you very much.
Thank you, Dr. Ali. Yes, currently there are various trials to include the AI and machine learning, especially during the optimization process, quality by design, the incorporation, even different modeling techniques, the computational models. I think these are one of the future directives that have been advised by many nanoscientists. Some of them, they are using these tools to, for example, to optimize their nanoparticles. But most of the recommendations nowadays, especially during the optimization steps, is to use the quality by design and all of these ML, the models, the computational, even before starting the experimental work. Yeah.
Okay.
Dr. Abdullah?
Thank you very much, Dr. Rami, for this nice presentation. I am Dr. Abdullah. I'm a microbiologist, and my question is just a rather a broader question, not about mycetoma specifically, but about different microbial agents and different microbial targets. Is this nanoparticles have anything to do with the entry of the antibiotic or the antifungal inside the cells? And does it promote the avoidance of the efflux pump with this antibiotic or antifungal get inside the cells? And also, does it provide any protection against degradation? You know that many antibiotics and antifungal can be degraded by certain enzymes produced by the microorganisms. And my last question is, does it affect the binding to targets? For example, the beta-lactam antibiotics, we normally know that it's go and bind to the penicillin binding protein in different bacterial cells. Does this type of nanoparticles have anything to affect the nature of the antifungal or the antibacterial in binding to the targets? Thank you very much.
Okay, thank you, Dr. Abdullah. So if I got you, so you are speaking about the interaction of the antibiotics with the pathogens, and does it affect if they are loaded into these nanoparticles, and the permeability inside the grains, and what other question, Dr. Abdullah, sorry?
As I told you, the first thing, as you said, the permeability and the entrance of this antifungal inside the particle inside the cell, whether it's a fungal cell or bacterial cell, and also does it affect the efflux pump of the antibiotic outside of the cell after it gets inside? Does it still be binding to the antibiotic inside the cells or not? And the last two questions about the protection against the degradation and the effect to the binding of the targets.
Okay, so mainly the nanoparticle, they are working as a carrier more than a complex nanoformulation, meaning that these nanoparticles, they will deliver the drug to the site of action, and then at the site of action, they have the capability to release the antibiotics itself. And then the antibiotics, if they are working or they are efficient, they have to interact with the pathogen itself. So mostly they are not working as a drug nanoparticle complex with the pathogens, more than these nanoparticle will promote the delivery of these antibiotics to the site of action, and then at the site of action they will release the drug, and then they should work directly or interacting with the pathogen itself. So, for example, as you said, if there is any degrading or some resistant report due to the releasing of some degrading enzymes to the antibiotics, or if there is any mutation at the receptors of pathogen, I think here we need to think about another approach, because the nanoparticle at this approach, it will not work. Here, our proposed approach is to overcome the grain, or for example, for other infectious disease, is to overcome the barrier of permeability and then releasing of these antimicrobial to work directly with the pathogens. So this is with the hydrolysis, if there is any enzymes. With the permeability, as we said, they can overcome different or various hurdles related with the permeations, because the nanoparticle itself, they can penetrate using various approach via the cell, intracellular, or between the cells, or using different receptors, engulfing of these nanoparticles. For example, as you know, the macrophages itself, they can't, you can't, for example, deliver the antibiotic inside the macrophages. But sometimes when you encapsulate your drug within the nanoparticle, you can target these macrophages, especially, for example, in the especially in the MTB, because as you know that the MTB is encapsulated within these macrophages, so you can target them for eradication.
You are correct. For intracellular parasites and bacteria, it's good molecules because it will be engulfed by the macrophage and will be inside the macrophage itself. But the issue is that in mycetoma, we have a very vascular lesions, and we are assuming that the drug concentration at the site is really high. But my question is, what induced the release of the antifungal or antibacterial at the site of the infection? So how the nanoparticle will know that this is the target of the infection?
Okay, that's one of the parameters that you have to optimize or to design based on the selecting of the nanoparticle itself. Some of these nanoparticles, they can release the drug based on the different environment, pH, for example. For example, sometimes you can design the nanoparticle and they behave differently at different pH, or sometimes at a particular enzymes, they can disrupt or they can dissolve to release the drug. Sometimes they can be stimulated using external stimuli like the heat, the magnetics. It's based on the disease itself. But mostly these nanoparticles, they can release the drug slightly based on different microenvironments of the disease. For example, such an example, you can propose one of these nanoparticles for the macrophages. We know that the pH at the macrophages is slightly acidic compared to the blood, for example. So we design these nanoparticles containing the drug. Then these nanoparticles inside the macrophages, due to the difference in the pH, they can behave in a different conformational, and they can release the drug better or in a high percentage compared to the blood pH, for example.
Okay, in case of mycetoma, most of the mycetoma agents produce grains which contain cement material that protects from the fungal, the antifungals, antibiotics. But if there is any way, for example, to formulate these nanoparticles to be affected by the concentration of the zinc, for example, because we know that the zinc concentration at the grain side would be at a higher concentrations.
I think we need to assess these environments. We need to study the microenvironment inside these grains, if they can be stimulated with this zinc high concentration, or the pH if it is different, or if there is any variations in the enzymes, or there is any enrichment in some enzymes or material that can change the behavior of the carrier inside this grain. So it needs some literature evaluation.
Thank you.
Okay, Dr. Rami, before we move from this point, you've highlighted that the nanocarriers serve as a carrier. But then, for example, in the case of itraconazole, it's metabolized in the liver by the CYP3A4 enzyme, and it's converted into an active metabolite that is hydroxyitraconazole. So while does the itraconazole can reach the liver to be metabolized by the CYP3A4 enzyme?
So if the drug is prodrug and it need to be converted to the active metabolite, then these drugs should be released from the nanocarrier before reaching the site of actions. If we load this drug in these nanoparticles, it will not be exposed to the metabolic enzymes, and they will be protected inside this nanocarrier or nanocomplex. And I think it's the better way is to use the active metabolites of the drug directly, especially if we are using a targeting behavior for the nanocomplex or nanoformulation.
But in the case of itraconazole, both the itraconazole and its metabolite, they have some varying degrees of activity. So they're both active. The itraconazole itself is not a prodrug.
Both of them, even for the itraconazole, it has some potential activity.
Yes, yes. Both
Okay,
the itraconazole and the metabolite.
And the metabolite, both of them.
Yeah.
Okay, yeah. So then we can use it, yeah, directly.
Okay, thank you for this. Lemise?
Hello, and thank you so much, Dr. Rami, for this wonderful presentation. My question is regarding the stability of the nanoparticles themselves, and also the route of administration. Because we know that, for example, the majority of mycetoma patients live in rural areas that they don't have access to. For example, they cannot keep the medication refrigerated if it's not heat stable. And also, one of the issues that has been a major hurdle in using liposomal amphotericin B is the need for it to be administered intravenously. So this means that the patient has to be hospitalized, and of course, this will add on to the expense of the treatment. So I guess my question, what type of routes of administration can such nanoparticles be administered by? Are there any like oral nanoparticles? And also the stability of these nanoparticles, given that most probably they will be stored in a place that there are no electricity or refrigeration.
Okay, thank you. So for the stability, for the nanoparticle as general, nanoparticle can be formulated using different dosage form and route of administration. I think most of the clinical trials people, they are just going to the parenteral way because it's a more easiest way to evaluate compared to the other routes. And also it depends on the location of the disease itself. For example, for the respiratory diseases, most of the people, they are now just looking for the pulmonary roads, for example, like the aerosols. For the diseases that in the systemic circulation, they just looking for the parenteral one. So I think for the mycetoma, my proposed route of administration could be the parenteral one, but at the same time, it's a long journey. So it's not like now we can say, we can try, for example, the oral one, and then assessing the bioavailability compared to the parenteral. But for example, the parenteral, you can achieve the higher concentration with lower drug quantities from the dosage form. In terms of the stability of the nanocomplex or nanoformulation, also it depends on the drug itself and the material of the nanoformulation, but they can be stored at room temperature, for example, if they are in a dry form, especially when we are using most of these antifungal or antibiotics. But we are just changing their behavior from the classical powder into these molecules loaded into nanocarriers. Most of the polymeric or most of the lipidic nanocarriers, they are stable at room temperature, so they can be stable. They can be stored at room temperature. But at the same time, a lot of stability studies need to be done after developing the final dosage form.
I guess my question, then like an additional question, would a nanoparticle that is used parenterally be the same nanoparticle that would be used orally, or would they be the same? Because if they're different, then it means that you're putting a lot of effort and resources into developing something for the parenteral route, which we know is challenging to use in mycetoma patients, because as I said, amphotericin B, liposomal amphotericin B is an example. And one other thing, I think when we talk about room temperature, we think about 25, 26 degrees centigrade. And we know that in these rural regions and in our countries like Sudan, this is definitely not the room temperature. It's going to be much higher. You can say like 30, 35, 40, depending on the season and so on. So would they still be stable?
Even for the classical, Dr. Lamis, even for the classical conventional dosage form, they are written that the room temperature of 25, and I think most of the people, they are storing these conventional dosage form in a higher temperature. So the situation in Sudan is different. So yeah, we have to look about the suitable storage temperature, even for the classical or the conventional dosage form. When you ask me about changing of the dosage form, I think when you are submitting your documentation for the clinical trials, for example, if you want to change, for example, the amphotericin B loaded in a nanosystem for parenteral way, and then you want to change into another route, you have to submit based on the clinical trials rule. You need to submit a new application if you change the route of administration for the drug. So I think for the clinical trials, you have to select the route of administration from the beginning, because changing the route of administration for the same drug, it will be considered as a new drug.
Yeah, so this is exactly my point, is maybe it would be good from the beginning to look for a nanoparticle that could easily be administered via the oral route, so that you don't go through the whole process, which we know is very tedious and time consuming and costly, for a drug to finally only be available in a parenteral route. This is just a suggestion. Thank you.
Yeah.
Thank you, Dr. Rami.
Sabir, Dr. Sabir.
Thank you. Thank you, Dr. Rami, for fruitful presentations. I have a question, and I expect the nanoparticle like a chance for the future delivery for the topical. If am I right? Could the nanoparticle be like topical gel or cream for treatment of mycetoma, specifically the fungal one that need the surgical operation? The second question that I have is, can we design and discover a multi-target in order to have a synergistic effect, and then we use a nanoparticle to deliver that multi-target anti-mycetomal drug?
Okay, thank you, Dr. Tasabi. So what do you mean by multi-targets? So you mean that different possibilities within the grains itself?
Yeah, different protein. I mean, for example, we can target the grain and the melanin, for example, melanin pathway, and a different protein in a melanin pathway. At the same time, loaded that molecule with another molecule that they can target other protein like the protein of the cell membrane, for example?
Yes, there is a possibility you can conjugate more than one ligand onto the surface of the nanocarrier. For example, one for the melanin and one for the proteins within the matrix of the grain. So you can achieve multi-targeting behavior of these of the nanoformulation. Also, I think you asked about the topical capability to topical formulation or topical nanoformulations. Yes, you can achieve, you can select different types of vehicles, and then you can disperse your nanoformulation within these vehicles. So it's just similar to the classical one, but the difference now of the dispersion material, now they are in a size different from the conventional one. And I think there is one of the, if I'm not wrong, there is one of the approved FDA. It's one of the topical, so I think it's a topical formulation based on nanomedicine.
Thank you for this, Dr. Rami. Ala Mudawi, I see that you have a raised hand. Feel free to unmute yourself.
Hello, everyone. Thank you, Dr. Rami, for this informative session. I just want to ask, or I am wondering about the elimination routes or the possible elimination routes for nanoparticles in order to avoid the toxicity or the possible toxicity we are having now in those type of drugs. Thank you.
Okay, I think there are still some work need to be done because Dr. Ala said that, yeah, there are some toxicity related with some materials. So still there is a broad area for research or a gap area to evaluate the pharmacokinetic behavior of these nanocomplex, the extended residence time of this material in the body, and how these material can be removed and for how long they will stay. I think there are various ongoing research to evaluate the pharmacokinetic pathway of these nanocomplex, because some of them, they can be hydrolyzed. For example, one of the polymeric nanomaterials, the polylactic glycolic acid PLGA, they can be hydrolyzed from the normal or the biological enzymes to lactic acid and glycolic acid. So some of these nanoparticles need to be evaluated in terms of their accumulation, in terms of their pharmacokinetic pathway, and removal outside of the body. And before that, I think even in the in vitro, you will assess the toxicity of this material, the toxicity of the drug-loaded complex of this nanoformulation before to go further with the in vivo or the other clinical trials.
Okay, thank you, Dr. Rami. Question away from the preparation of the nanoformulation is that we know that mycetoma affects poor patients in poor areas. So what is the cost of these nanoformulations?
It's easy to approximate the cost, but for any new discovered material, as you know, the scientists, they are inputting a lot of money during the clinical trials, during the research, the development. So I think based on the discovery, it's similar to the discovery of new molecules. So at the beginning, especially during the patency period, the cost will not be it will not be a cheap drug, but I think it can be funded by the international organizations, for example, like the WHO or different organizations. But it will not, yeah.
With these formulations, I mean, be affordable.
It can, you can't, yeah.
Will it be affordable in a country like Sudan?
It's a long journey. It's a long journey. So you can't approximate the cost now in terms of the final dosage form. Yeah.
Oh, okay. But the already designed nano formulations, like for example, the doctor would be seeing, is it expensive?
So
the already drugs that are now used.
So the doxorubicin, I think the cost around $300. Yeah,
Okay.
I think so.
If anyone has a question, you can raise a hand or write in the chat. Yes, Professor Fahad.
Thank you very much. Just I want to say that quite a long time ago, we studied the vasculature of mycetoma, and we found that the arterial supply of mycetoma lesion is very much vascular, to the extent that having the same appearance of a tumor plush, indicating that the blood supply to the mycetoma, it is perfectly well, because this is a chronic granuloma. So that time we thought, why not to infuse the lesion regionally through a catheter with the drug, so as you can deliver a large amount of the drug to the tissues affected there. And nowadays, Rami is telling us we can do it through a nanoscience. This is amazing how we can see the people how to change their ideas and thoughts and experimental work. So that time, nobody thought about this nanotechnology that can facilitate the delivery of such a drug to the tissues at that time. So I think this is the progress in science, in the technology, and the progress in the delivering very good care for the patients over the years. Thank you very much, Rami, for us changing our mind from these very invasive catheters and the local delivery of the drug, and then to the use of this nanoscience and this technology.
Zulfa, I see that you have a raised hand. Please unmute yourself.
السلام عليكم ورحمة الله وبركاته. أتمنى أنكم كلكم تكونوا بخير وصحة وعافية، وكل عام وأنتم بخير، وربنا يعيد علينا وعليكم عواما عديدة من مديدة يا رب. أنا بصراحة عندي
Excuse me.
Yes.
Can you English please, because you have colleagues that cannot communicate in Arabic.
Yes, I was just like greeting you all. I am a third year medical student. I'm still like an undergraduate, and I actually was very interested after studying microbiology in the mycology and especially in mycetoma. And I want to ask you if you have any advice for me in how to study. Especially, I am intending to do a research in this topic, but I feel like mashallah, like you, Prof. Fahad, have made many achievements in this topic. I want to ask you for your advice. What do you think I should do as a medical student and as a future doctor to study and to do further in this topic? Thank you so much.
Thank you very much. We have the Mycetoma Young Researcher Association, and I would like to invite you to join us and to be active part at this association.
Thank you so much. Thank you so much. Was a very informative session, and I like it so much. Greetings to all to you.
Thank you. We're happy to have you. I've just written my email on the chat, so you can email me directly, and I'll let you know how to join the Mycetoma Young Researchers Association. Dr. Abdullah, I see that you have a raised hand.
I'm just asking Dr. Fahal about, have you ever tested the slow release or a modified release of this antifungal or antibacterial for mycetoma?
Sorry, the voice is interrupted.
Yeah, I'm sorry, Dr. Fahad. I'm just asking about the modified release or the slow release of antifungal or antibacterial agent for the treatment of the mycetoma. Do you have any experience with that?
No, not really, not really.
Yeah, because we have been thinking about the future in the past. We have been thinking about the concentration of the drug in the tissue, but as you said, the nature of the lesion is quite vascular, and we are sure that the concentration of the drug or the different drugs as the lesion side is really high. But the question is, do we need to have like a continuous infusion of the drug over a time in order to achieve better effect? And I think we can try to test this in an vitro model as well to see whether the continuous exposure of the antifungal or antibacterial, does it make change or does it help in the penetration of the antifungal inside the grains, inside the fungal mycelia? Because this is actually the most crucial thing. It's not for me, it's not the release of the antifungal or the direction of the antifungal to the side of the infection, because as we know that mycetoma have a very good blood supply. But maybe if we try to make a continuous release or a modified release of these drugs, and we can try to test that maybe in as I said, in a vitro model, maybe it might make some difference.
Yeah. Yeah, it is a good idea. Eh? Yes. As you know, vosoronozole have a long half-life, and that is why you have it once per week after loading the drug for 3 days, and then after that you continue with this high level of the drug in the serum and in the blood for that.
Yeah, perfect.
Okay.
We're 15 minutes.
Yeah.
Past
Okay.
our time. The discussion, it was grateful. So, Professor Fahal, if you would like to add anything before we end up our session.
Yeah. I think Rami had gave us a very excellent overview about the use of these nanoparticles and nanoscience in general. And I think this is an area we need to work hard on it. And we need to utilize technology. And we need to utilize even the AI with this technology to achieve something for our patients, for the diagnosis, the treatment, and even for the prevention of the mycetoma and other neglected tropical diseases. The good thing about this session that it is the start of collaboration between our center and South Africa universities. And again, this is an example of a South-to-South collaboration, which is more important than the other collaboration. We need to work together to address our problems and to find a solution for our local community and the local problems we are facing dealing with the patients and the communities. And thank you very much, Rami, for this nice, excellent presentation. And we're looking forward for a great collaboration between our institutes. And thank you very much.
Thank you again for for these invitations. Thank you all.
So by coming to this point, I'd like to thank you all for attending today's session. And Dr. Rami, thank you so much for this eye-opening session. And we look forward for further collaborations, of course. And we were really happy to have you all here today. And until we meet again in our next session, I hope that you have a wonderful time. Thank you all for joining, and goodbye.